Okay, girls. So there are only 4 of them who are still giving the exam, so they'll quickly join us, inshallah. So let's start our topic, enzymes. Let's quickly recap the contents we covered in the last lesson. All right, as you all know, this is chapter 5, and today we are going to cover, let me tell you the page number. So today we'll be doing page number 52, inshallah, about the properties of the enzymes. All right. So we'll start with this verse, which is related to the enzymes about the honey. So according to Allah, we should consume a lot of honey because it contains healing for the people. Indeed, that is a sign for people who give thought. Meaning, if we are following the Quran and Sunnah, then we have many signs like that which Allah has given us to follow to benefit from them. Right?
Yes, I just thought you are sharing the PowerPoint.
I'm sorry, sweetie.
Are you sharing the PowerPoint?
Yeah, I'm sharing. You can't see it?
No.
All right, I'm so sorry. Thank you.
Me too. Miss, I can't see it.
You can't see it. Okay, thank you. I'm so sorry. When I'm shifting from this channel to another one, I believe the sharing option is getting disabled. Now, can you see it?
Yeah.
All right, great. Thank you so much. Okay, so this was the verse which we just spoke about. So the objectives for today's topic would be to learn as to how enzymes are affected by temperature and pH. So far we studied as the function of the enzymes as to how do they function as catalysts, and how do they help in speeding up the reactions, and how does they come into contact with the substrate molecules and all. So today we are going to study about the objective as to how enzymes are affected by the temperature and pH, and why enzymes are affected by the temperature and pH, inshallah. All right, under the KWL, let me ask you the questions related to the topic we covered yesterday. Yes, any questions or doubts? Who's about to talk? These are the ones we covered the day before. Yeah, so yesterday we were discussing about a substrate. Who's going to tell me what is a substrate? You can read out from the slide here.
A substrate is the molecule, or it could be a protein or anything, or a carbohydrate in which an enzyme acts.
Very good. The enzyme acts on the substrate molecule either in breaking down the carbohydrates, proteins, or fats, or on the various molecules which are present in the liver, pancreas, and all this. And what is an active site? So this region where the substrate molecule comes and binds to the enzyme is known as the active site. Near the active site, the molecule, it binds to the enzyme so that it forms an enzyme substrate complex where the reaction takes place and the products are released. All right, so this is what we studied about the substrates. You can also define them as molecules that are the molecules that react in the enzyme in a catalyzed reaction are known as substrate, and the molecules that are produced are known as the products. Okay, so different enzymes, they have specific function in different types of reactions. Not all enzymes can work on all the reactions, as we studied yesterday. We also studied about how the name has been given depending upon their function to various enzymes, and each enzyme, it ends with ASE, except for a specific enzymes like pepsin and renin and trypsin, which have been named originally. Apart from that, all the names, they end with ASE. You can recognize the enzyme. So these are the 5 properties of enzymes. And we also studied that each enzyme has a specific job as to how they can carry out only the specific job, meaning they can only work on the substrate molecules which fit into them like this. Okay. And And so this is what we study. This is the active site, and this is how the substrate comes and fits into the active site. And the enzyme, it works like, and the substrate, they work like a lock and key mechanism. As the key fits into the lock, this particular substrate can only fit into the enzyme, and only then the enzyme can catalyze the reaction. We also studied with this example yesterday as to how the substrate, it fits into the enzyme and how does the reaction take place. Can someone explain the 3 steps to me, how it happens? It looks like a large bobbly nose, right? The enzyme. Okay, who's going to talk about it? Who's going to explain the 3 steps?
I don't think that we took it till here.
We took it till, I remember taking this slide yesterday. Isn't it, Shahad? Did we take it? Laila, Shahad?
Yes, Miss, we took it.
Yeah, Miss, we took it.
Yeah, we took it, sweetie. We were doing this slide, and then I was about to assign the homework, but I said I'll do it the next day, inshallah.
Miss?
So can you do it, or do you want me to do it?
Miss, can I ask you a question before you explain?
Yes, sweetie, go ahead.
Miss, so are the products that are produced from the enzymes, are they a broken down version of the substrate, or are they new substances that are produced?
Sometimes they are broken down versions of the substrate itself, always, unless there is a reaction which changes them. For example, if you're taking an example of the glucose being broken down, okay, so during the respiration, the glucose is broken down into carbon dioxide and water, isn't it? And energy is released. So in this, the products, of course, they are from the glucose itself, but they're a little different. Whereas when you're breaking down the starch in this example, it is breaking down into the products which are disaccharides, meaning the glucose units itself, or starch, it is a long chain made up of glucose units, isn't it? So once it is broken down, it will break into single or double glucose units like this. Clear, Shahad?
Yes, Miss, thank you.
Yeah, so sometimes it is the same product whenever we are talking about, for example, the proteins are broken down. They will break into amino acids, and especially fats. When they're broken down, they'll break into glycerol and fatty acids. So they will break into the simplest form of the same substrate substance. Clear?
Yes, Miss.
Yes, sweetie. So who's going to explain this? Quickly, we are wasting time, or shall I explain it?
Miss, can you explain it, please?
All right, sweetie. So this is how the substrate, it comes and fits into the enzyme. So this big thing which you see over here is the enzyme. For example, over here in this example, this is amylase enzyme. So amylase, it works on what? It works on the starch, which is a carbohydrate. So this starch now, it comes and fits into this active site of the enzyme. Okay, so it has to fit in exactly like the key fits into the lock over here in the active site. So once this fits in here, it forms an enzyme substrate complex, and now the reaction starts. Okay, so the first step is the substrate molecule, the starch in this case, it slots into the active site, meaning it fits into the active site like this. Then what happens next? Each enzyme has an active site into which its substrate molecule fits in exactly. As you can see, it needs to fit in exactly like this. And this enzyme is amylase, which we are talking about. And its active site, it is just the right size and shape for the starch molecule. Meaning this amylase enzyme, it can only work on starch. It cannot work on proteins or lipids or any other substance. It is made only to break down the starch molecules. Okay, so that is why they say each enzyme, it has a specific function. It cannot work on all the types of reactions. And it has a specific shape into which only the substrate molecule can fit. So in this enzyme amylase, only the starch substrate can fit like this. So now the reaction is taking place. So the starch is broken down by the action of the enzyme amylase, and the products are released. So what are the products? The starch is split into maltose molecules. The maltose, which are the disaccharides, right? So this starch molecule, it has been broken down further into maltose molecules, and the enzyme is unaltered, meaning we studied in the definition itself that though the enzymes, they help in speeding up or catalyzing the reaction, the enzymes themselves, they will not change, isn't it? So over here, the enzyme is unaltered, and it is ready to accept another part of the starch molecule. Now this starch molecule has been broken down and the products are released, and the enzyme is ready to accept another starch molecule, because in the food we eat, there are a lot of starch molecules which we eat, isn't it? For example, if you're eating rice or wheat also contains gluten, which is a starch as well. All right, so this starch, the next starch molecule now comes and fits into the enzyme, and again it is broken down into simpler substances like the maltose molecules. Clear, sweeties? Any questions?
No, miss, thank you.
Clear?
Yes.
All right, the same picture you have on page number 51 in your textbook. So with this we are done with 5.1, which is biological catalysts. Okay, 5.1 topic is done. So you have questions on page number 51 from 5.1 to 5.6, which will be the homework for today, inshallah. You may complete it over the weekend, and also don't forget to complete the MLG assignments, which I uploaded. You may do it over the weekend when you find time, inshallah. All clear so far? Shall we move on to 5.2, which is properties of enzymes now?
Yes, miss.
All right. So we are on page number 52 now. We are going to discuss about the factors or the properties which affect the enzyme. So today's objective is to study as to how the temperature and pH affect the rate of the enzyme. Okay, rate of the chemical reaction. So the rise in temperature increases the rate of most chemical reactions. Okay, the greater the temperature, the faster the reaction is. But again, not very high temperatures. Okay, so a rise in temperature, it increases the rate of a reaction, and a fall in temperature, it slows down the chemical reaction. However, the temperature, it should not be above 50 degrees centigrade. Because if the temperature is above 50 degrees, it is too high for the enzymes. The enzymes, what do they happen? They get denatured, meaning their shape, it changes and they stop working. Okay, so we can say the best temperature or the optimum temperature for the enzymes to work is somewhere between 10 degrees centigrade to 50 degrees centigrade. Okay, so neither the temperature has to be very low nor it needs to be very high. It has to be the exact temperature, which is known as the best or the optimum temperature for the enzymes to work. So what happens when the temperature is above 50 degrees centigrade? I will also show you. So above 50 degrees centigrade, the substrate molecules, they vibrate. What happens when we are heating something on the stove? When we are heating, the molecules, they start vibrating, isn't it? So if we are heating it above 50 degrees centigrade, then the enzyme will get denatured or lose its shape, and of course it cannot carry out its function. Okay, so this graph over here, it explains as to how the increasing temperature has an effect on the rate of the reaction. Okay, so it shows the effect of increasing temperature on the rate of an enzyme catalyzed reaction. So on the x-axis, you can see the temperature increasing from 10 to 60, and on the y-axis, you can see the rate of the reaction as to how it increases. Can you see how it increases along with the increase in temperature, and it falls down once it is above 50 degrees. Okay. So you need to remember that above 50 degrees centigrade, the enzymes, they cannot work, they get denatured, or meaning they lose their shape. So this is how the denaturing takes place. At higher temperatures, it speeds up the movement of the substrate molecules so that when they collide with the enzyme, they have more energy and are more likely to bind to the active site. Okay, so this is how the temperature, it affects the enzymes. So whenever the temperature is more, it helps in speeding up the reaction. How? The substrate molecules, they start vibrating. They come and collide with the enzyme with more energy, and so the reaction takes place more faster, at a more faster rate. But again, not at very high temperatures. Okay, so the enzyme molecules themselves also gain in energy as the temperature rises, so that they begin to vibrate. So when the substrate molecules start vibrating, even the enzyme, it starts vibrating, isn't it? So eventually the enzyme molecules vibrate so much that they become denatured, they lose their three-dimensional shape and can no longer bind to their substrate. So this is what happens when the temperature is too high, above 50 degrees centigrade. The enzyme, it loses its shape, its three-dimensional shape, and it gets denatured. So it cannot carry out its function anymore. Okay, so because of this, the high temperatures, they reduce the enzyme activity. So what is the solution now? So what is the optimum temperature for the enzymes to work? Now what is optimum temperature? It is the right temperature or the best temperature at which the enzymes can work best. So each enzyme has an optimum temperature, which is a balance between the two effects as shown in the graph below. Okay, so the temperature, it neither should be very low nor should be very high. It should be the optimum or the exact temperature for the enzyme to work. Now this temperature is different for different types of enzymes. For example, the human enzymes, meaning the pepsin, the salivary amylase and the lipase, all of them which work in the humans, in our stomach and the intestines, liver, pancreas and everywhere. For those human enzymes, the optimum temperature is our normal body temperature. What is our normal body temperature, girls?
37.
Yes, 37 degrees Celsius or 98.4 Fahrenheit. So at this normal body temperature, the enzymes will work best. Okay, so this is the optimum temperature for all the human enzymes. Whilst for the plant enzymes, the optimum temperature is rather low. It is around 25 degrees centigrade. All right, so the optimum temperature is different for different types of enzymes depending upon the location where they act upon. Maybe in humans, in the stomach or pancreas, or maybe in plants, depending upon where they act, the optimum temperature is different. So clear about the optimum temperature, girls? Any questions? How do you define the optimum temperature? What is the optimum temperature? Can we say it is the best temperature at which the enzymes work best?
Yes, Mouse.
Yes. So what is the optimum temperature for human enzymes?
37.
37. And for the plant enzymes?
25.
25 degrees centigrade. So that is why you will realize that whenever we get fever and the doctors say that your temperature, it should not be above 103. Okay, so at that high temperatures, our metabolic reactions, they cannot be carried out. Why? Because the enzymes, they get denatured at very high temperatures. Okay, so for our body to maintain to function properly, it needs to have the normal body temperature of 37 degrees Celsius or around that. Okay, so at very high temperatures, that is why we get, when we have very high fever, we get headaches and vomitings and diarrhea, and the various things might escalate if we do not bring down the temperature immediately. So we should not, it is very dangerous for the temperature to rise above 101 or 102. Of course, it happens sometimes if you have an infection or a cold or a flu or anything, but we need to bring it down back to the normal immediately. So this is how the homeostasis also works in our body. The various systems, they function together in order to regulate the body temperature. This is how the homeostasis, the constant body conditions are maintained in our body. Okay, so how do you define optimum temperature? Is the temperature at which the maximum rate of the reaction occurs is called an optimum temperature. It is also known as the best temperature for that enzyme. Okay, so here are some examples of optimum temperatures. The fungal and the plant enzymes, they approximately work at 20 degrees centigrade. That is their optimum temperature. The human enzymes, they work at our normal body temperature, which is 37 degrees Celsius. And some of the enzymes produced by bacteria, the bacterial enzymes, for use in the industry. You know, the bacterial enzymes are used a lot in the food industry these days, which are the beneficial bacteria, and they can work best at 90 degrees centigrade. So that is a temperature at which they can work best. All right, so these are the various examples, and this is how the temperature, it affects the enzyme activity. Clear, ladies? Any questions?
No,
You may ask me if you have a doubt or question or anything. All clear?
Yes. Yes, Miss.
All right, sweeties. Then please complete the homework on page number 51, which is from question 5.1 to 5.6. And also don't forget to complete your MLJ assignments. Okay, Ahad, do you have a question?
Yes, Miss.
Yes, sweetie.
Teacher, if the enzyme is denatured, can it come back to its original shape?
No, it cannot. So once it's denatured, it loses its shape. So that's it. It cannot come back to original shape, because as and when the new enzymes are being secreted by the body, the next enzyme will carry out its function. It will be denatured, meaning those protein molecules will be used up in making of new cells. As the protein synthesis takes place and new enzymes are formed again. Clear ahead?
Yes, Miss. Thank you.
All right, sweeties. Then you have a relaxing weekend ahead. Okay, let's meet next week, inshallah. Enjoy your weekend. Stay safe and have a lovely day ahead.
Thank you, Miss.
Bye. You're most welcome, sweeties. Bye-bye.